Hello students, now that we are well aware about what plant growth is all about, we have to deal with plant growth regulators. In common language, we term it as plant hormones. Simply, we just give them the terminology of being a hormone. Why? Because they are organic compounds which are synthesized at one place and through some mechanism are transported to their site of action. Same as in the case of hormones. So we believe them to be hormones, but they are not hormones typically. They are, it is a better way to address them as plant growth regulators because they cannot be termed as hormones just on the basis that we say that they are synthesized at one place and they work somewhere else at their target organ. So we deal here with the various organic compounds which have a crucial role in the plant growth and their developmental processes. So we are going to deal with plant growth regulators. When I say plant growth regulators, then there are 5 growth regulators you must be knowing. First one is auxin. You might have heard about these terms. Auxins. Then the second class is of gibberellins. Third ones are cytokinins. Then there is a famous hormone, stress hormone, that is abscisic acid. And then ethylene, which is a gaseous hormone and is responsible for ripening of fruit. Whenever you are tasting a very juicy and fruitylicious fruit, that means it is a lot of ethylene has been involved in its ripening because it is the gaseous hormone which is responsible for growth and ripening of fruit. So we would deal with it individually. We are going to deal with each of the hormones. First of all, we deal with auxins. So what are auxins? Before we start anything, we need to know that what do we study when we study about plant growth regulator. First of all, it has to be discovered somewhere. So we do put a little bit of stress on how that particular hormone, how and where in fact it was discovered. Then we come to the point that there are certain techniques in identifying the hormone that is known as bioassaying. I'll come to the term when we are sure where that hormone is located or the regulator is located, we study its effect on the plant growth. So there are 3 things which are to be studied: the discovery, the bioassaying, and their role in the plant growth basically. And of course we deal with their chemistry as well, little bit of chemistry just for understanding purpose. So one by one we are going to deal with each of the hormones, and primarily you have to remember the function that they are playing, and you have to remember one more thing, that which hormone is antagonistic to the other one. When I say antagonistic, that means if one hormone is leading to the increase in length, there would be another hormone which would be working antagonistically, or in simple terms oppositely to that particular metabolic function which one hormone has conducted. So let's see what auxins are all about. Auxin is the first hormone that was discovered. It was discovered in human urine. Okay, so the scientist who discovered it was Charles Darwin. He discovered it in the human urine, and one thing might confuse you, that how can a plant hormone be present in human urine? We ultimately depend on plants for food, so that in that way it is bound to be found in the urine, and from there it was discovered that there is a plant growth regulator which might be having some effect on the plant growth, and that happens to be auxin. Now as I told you, we need to see its bioassaying and we need to see where they are present, how do they work. So the major site where you would find the auxins is the shoot apical meristem. Now we know that there are 2 apices in the plant. One is shoot apex. The plant is like this. There are roots beneath the soil. This would be the root apex, and over the top this would be the shoot apex, where you would see the plant rising forward. The growth is shown downwards and the plant increases in the height. So the shoot apical meristem would be having the auxins. Now how it was discovered? It was discovered by an experiment and a bioassaying process. Now I used this term earlier. Now you need to understand what is bioassaying. Bioassaying is the process in which you find the potency and the concentration of a particular compound and their functions. Alright, you just analyze where that compound is present in how much quantity, and after that you make sure that what are its functions. So bioassaying was done by F.W. Wendt. He was also a botanist. Like Charles Darwin, discovered the auxins. He was the one responsible for telling us that how the auxins are, where the auxins are found, what is their concentration, highly concentrated at the shoot apical meristem. Another site of finding the auxins would be the embryo of seed and young leaves. You see, I am telling you as I use the term shoot apical meristem, embryo of the seed and young leaves. We know that maximum growth in a plant would be shown at these sites. So this particular hormone, that is auxins, group of chemical compounds, they would be termed totally as auxins. Those, one thing I need to make clear before I go any further, you must keep in mind that if I say a human hormone like thyroxine, so that would be one hormone, one chemical compound that I would be referring to. If I am referring prolactin, it would be one chemical compound, but in case of plant growth regulators, auxins would refer to all those compounds, all those organic compounds in the plant, naturally synthesized or synthetically synthesized, which would show same function in the plant physiology. Okay, the morphological functions or the those metabolic functions which would be occurring inside the plant would be similar, and whichsoever chemical brings that similar function would be termed as an auxin. This is the difference between plant and human hormones. So you can find the auxins in shoot apical meristem. You'll find them in embryo of the seeds, you'll find them in the young leaves, and as I told you in the beginning, they are synthesized at one site and then they are moved to the other site. So they would be moved by polar transport, and that polar transport would be unidirectional. Now when I use the term polar transport, it means it would be according to the density gradient. Wherever the gradient would be more or wherever it would be required, the auxin or the particular compound would move according to the polarity basically, and that polarity has to be unidirectional. Now you understand what is unidirectional. It means that it has to go in one direction, it cannot come back. Okay, so they are very much target oriented. Now I told you that they are naturally formed or they are synthesized synthetically. So natural auxin is indole acetic acid, IAA. Alright, and few of the synthetic auxins, you have to remember these names just by cramming it. Indole acetic acid is the auxin which is naturally synthesized within the plants, and over time horticulturists and other agriculturists, they have synthesized auxins synthetically as well, in which we have NAA, that is naphthalene acetic acid and 2,4-D, and it is a herbicide. Alright, herbicide means it kills the herbs. So when we are talking about lawn mowing and getting rid of the weeds, we do throw these auxins, synthetically synthesized auxins like Agent Orange. 2,4-D is also known as Agent Orange, so it would be thrown in your garden and the garden would get rid of the weeds. It is used as a herbicide as well. These are the various features of auxins that you need to know and remember. Now one thing I need to bring to your notice is that when there is higher concentration of auxins, it inhibits the cell elongation. Okay, why it inhibits the cell elongation? Because it favors the function of ethylene, which is a cell growth inhibitor basically. Okay, it is the growth inhibitor. So higher concentration of auxins would be as similar to that of the function of ethylene. Please remember it, it would work as similar to the function of ethylene when the concentration of the auxins would be higher. So we need to make sure that the concentration is also limited and restricted so that it does not bring about the similar functions that ethylene would perform, and cell elongation would be inhibited by that. Now understanding the basic functions of auxins, we need to know that what auxins do, because they are present in shoot apical meristems, apices in the plant. So they are going to bring about the lateral growth would be suppressed and they are going to bring about the vertical growth. First thing that you need to remember, there is a term known as epical dominance, which tells that the plant would grow vertically higher and higher and it would suppress the lateral growth, and that is the function of auxins basically. In function with cytokinins, which is another group of hormones responsible for cell growth, that is plant growth, ultimately resulting in plant growth, these cytokinins, they promote the differentiation of callus cells. Now when I use the term callus cells, I need you to understand that it is somewhat similar to the meristematic cells, which would divide and give rise to other plant organs in later on stages. So differentiation, that means giving rise to the cells, and their differentiation, that means their performance, the performance oriented, or they get accustomed to whatever function they have to perform, that process is known as differentiation. We have discussed about it, what a cell differentiation is, into root initials in the base of cutting. So supposedly you are having a rose stem that you are going to plant in the soil. You know how rose is vegetatively propagated with the help of a stem, and wherever you are having stem cuttings, root initiation or the root apices would be formed by the auxins basically. So that process of root formation in the rose stem is conducted by auxins along with cytokinins, not to forget. When there is a higher concentration of auxins, the apical bud of the shoot suppresses the growth of axial shoots, and this thing is known as apical dominance. Now, what is apical dominance? That the axial shoots, that is the lateral growth shoots, would stop their function, and not stop exactly, they would reduce their growth, and the main apex would rise further. That we discuss in the case of conifers. Conifer growth is somewhat like this, and over here you would see in conifers which are found in mountains and all, the apical growth is much dominant as compared to the suppressed growth of axial shoots. Axillary shoots which show the lateral growth, they will not be allowed to grow as much as in the presence of auxins. That is, pinching out of the shoot would take place. That means the topmost is growing further as seen in the conifers. Pinching out over here refers to conifers. You can relate both of these. And then the next step is the movement of auxins to shaded side of stem causes the cells to elongate so that plant grows towards the light, and this is known as phototropism. I want you to concentrate over here. I want you to read this and then understand what basically phototropism is. Now, when I talk about phototropism, it is somewhat responsible for bioassaying of the plant cell and understanding where the auxins are present and how do they function. Now, tropism is an important mechanism that is observed. You know what is tropism? Tropism is the behavior of plant towards a particular stimulus, that how a plant would react if a stimulus is provided. So when I use the term tropism, that means there would be some stimulus, some sort of stimulus, it could be light, it could be water, and the plant would be showing particular tropic movement towards that stimulus, and that would be termed as tropism. And when we talk about auxins, we deal with phototropism. Now, you know, light is an important factor for plant growth, so we talk about phototropism in case of auxins. And when we have to understand phototropism, we take into consideration this particular aspect. Now, what this is all about? This over here is the plant shoot tip, apex of the shoot, and you see when the shoot apex is having higher concentration of auxins, one thing you have to remember, that auxins always move away from light, sunlight, okay. So if you can see over here, when the direction of the sunlight is tilted, the direction of the sunlight being tilted, the auxins have moved away. That means they have moved towards this side, and over here we see cell elongation. Now, this is quite confusing. You might be having an idea, or you might not be having an idea, that this thing does not explain that how cell elongation is taking place. It is simply, what you have to understand over here is that supposedly we have sunlight over here, alright, and I know that there is certain compound which does not like much of sunlight, means it is not having affinity towards sunlight, so it moves towards the other side. The portion which is receiving the sunlight, auxin is moving away from the sunlight, and as we studied, what are the functions of the auxin? Cell division basically. So it would be concentrated over here. Now we see that this particular tip of the plant gets tilted. You can see over here as well, it gets little bit tilted. Why it gets tilted? Because the growth is taking place as this side. So if supposedly this side gets elongated to this much, obviously this side is not growing, so there would be a curve which would be occurring. This curve would grow further, and as visible on this slide, you can see it grows further and it shows little bit of curvature, and this is known as Avena curvature test, okay. Now, there was bioassaying done by F.W. Wendt. What he did in his discovery and bioassaying was that he took the seedling tip, he cut it and put some agar box over there, some little bit of agar was put over there. Now what happens is that because we know that auxins are located in the tip of the plant, basically shoot apex, these auxins move towards this gelatinous or agar gel, whichever is being used. This box, it gets this auxin. Now on cutting this tip basically and putting auxins over here, as you can see the auxin has been put over here, that side to which auxin is present, same way, the same growth takes place. That means auxin is somehow responsible for cell differentiation and cell elongation. I would repeat it again, that see, basically you are taking a seedling. It is not a seedling, it is a coleoptile which would further form a stem. You are going to cut it. Okay, when you cut it, you put some agar gel over there. Now this agar gel, because it is also somewhat gelatinous substance, the auxins would diffuse into the agar gel. Once they have diffused, the agar gel would have more and more of auxins. Now what is to be done, that this tip is to be removed. Alright, when this tip is removed, that gel box which had auxins in it is placed on that cut tip. Now because the auxins are gone, because the tip has been gone and auxins are present over in this gelatinous agar, they are going to diffuse into the plant. The chemical compounds they are going to diffuse into the plant and they would show the same curvature as shown by the process of phototropism when we understood it. In the previous slide you must be seeing this. This was what we see if we have to understand the function of auxins, and this is what we get to see if we cut the tip. That means somehow auxins are present in the tip of the stem. Shoot apices, their tip is having the auxin. So in this way we get to understand where the auxins are located, what is their concentration in that, and what are their functions. Next hormone that we are going to deal with is gibberellins. Now in case of gibberellins, there is an interesting story you need to know. Gibberellins are the hormones which are very important for plant growth. We are going to discuss the entire story of gibberellins, how they were discovered.